Welcome to an extraordinary journey into one of nature's most remarkable biological phenomena. In the coldest regions of North America, certain frogs perform what appears to be a miracle each winter. They freeze solid, with no heartbeat or breathing, only to thaw and hop away when spring arrives. Today, we'll explore the fascinating science behind freeze tolerance in wood frogs, spring peepers, and gray tree frogs. A survival adaptation so extreme it borders on science fiction, yet offers revolutionary insights for human medicine and cryopreservation technology. In the harshest winter conditions of North America, three remarkable amphibian species, the wood frog, spring peeper, and gray tree frog, demonstrate an almost unbelievable survival strategy. As temperatures plummet below freezing, up to 65% of their body water transforms into solid ice. Their hearts completely stop beating. Blood flow ceases entirely. All visible signs of life vanish as they become frozen solid for weeks or even months. By any conventional definition, these animals appear dead. Yet when spring's warmth returns, something miraculous happens. They thaw out and resume normal activities as if nothing extraordinary occurred. This freeze tolerance represents one of the most extreme physiological adaptations in the vertebrate world. While many organisms have evolved ways to survive cold, these frogs endure the formation of ice crystals throughout their tissues and organs, a condition that would be immediately fatal to humans and most other animals. This natural cryopreservation strategy has evolved over thousands of years, allowing these species to thrive in environments where other amphibians simply cannot survive. The remarkable survival of freeze-tolerant frogs begins long before the first ice crystals form. As autumn temperatures drop, these amphibians initiate a sophisticated biochemical preparation process. The wood frog's liver starts producing massive amounts of glucose, up to 250 times normal levels, flooding their bloodstream with this natural antifreeze. But glucose isn't their only defense. Urea and other specialized cryoprotectant compounds join this molecular shield. Unlike typical antifreeze that prevents freezing altogether, these compounds serve a different purpose. They allow ice to form in safe spaces between cells while protecting the cellular structures themselves from damage. This high concentration of glucose prevents excessive dehydration of cells as water is drawn out to form ice in extracellular spaces. Without this protection, the cells would fatally shrink and their membranes would rupture. The biochemical transformation is so extreme that if humans experienced similar glucose levels, we would be in a severe diabetic crisis. Yet for these frogs, this glucose surge represents not a disease state but a sophisticated survival mechanism refined over millions of years of evolution. The freezing of a wood frog follows a remarkably orchestrated sequence. It doesn't happen randomly throughout the body but begins in the extremities and gradually moves inward. As ice formation reaches the abdomen and spreads throughout the body, physiological changes occur in a precise order. The heart rate slows dramatically before stopping completely. Breathing ceases entirely. Neural activity becomes undetectable by conventional measurements. The frog enters a state that defies the traditional boundary between life and death. Yet at the cellular level, vital biochemical processes continue at an almost imperceptible pace, maintained by the glucose that saturates the tissues. Perhaps most astonishing is these frogs' ability to endure multiple freeze-thaw cycles within a single winter. Research has documented wood frogs surviving temperatures as low as -4 degrees Celsius, 25 degrees Fahrenheit, for more than 2 weeks. During the thawing process, the heart typically resumes function first, beginning to beat even while ice remains in other parts of the body. Within hours of complete thawing, these remarkable amphibians regain mobility and return to normal behavior patterns, with no apparent damage from their frozen state. The evolution of freeze tolerance presents fascinating evolutionary questions. This adaptation appears to have evolved independently in several amphibian lineages, suggesting powerful selective pressure in specific environments. The wood frog displays the most extreme freeze tolerance and has pushed the northernmost range of any North American amphibian, extending into the Arctic Circle in Alaska and Canada, territories that would be impossible to inhabit without this adaptation. Interestingly, freeze tolerance varies significantly even within a single species. Wood frogs from Alaska demonstrate superior freeze tolerance compared to their counterparts from Ohio or Pennsylvania. These Alaskan populations produce substantially more glucose and other cryoprotectants, allowing them to survive temperatures below -18 degrees Celsius, 0 degrees Fahrenheit, and remain frozen for over 7 months. This means northern wood frogs actually spend more time frozen than thawed each year, An extraordinary evolutionary achievement. This geographic variation suggests rapid evolutionary adaptation to local conditions. As frog populations colonized increasingly colder regions after the last ice age, they developed enhanced freeze tolerance mechanisms through natural selection. The genetic basis for these differences continues to be investigated, but research indicates both genetic adaptations and epigenetic changes contribute to the enhanced cold hardiness of northern populations. Freeze tolerance is also connected to drought tolerance in some species, as both conditions require cells to withstand significant dehydration stress, potentially explaining why particular species possess these abilities while closely related species do not. The ecological advantages of freeze tolerance are substantial. These specialized frogs can exploit habitats and hibernation sites that remain completely unavailable to other amphibians. While most frogs must hibernate underwater or deep underground below the frost line, freeze-tolerant species can simply hunker down in shallow depressions, leaf litter, or logs, even when these microhabitats will certainly freeze solid. This flexibility reduces competition for limited hibernation sites and decreases predation risk during the vulnerable winter months. It also allows these frogs to be among the earliest amphibians active in spring, as they don't need to journey from deep hibernation sites. They can emerge, mate, and secure resources before competitors become active. Wood frogs are typically the earliest frogs to breed in spring, often gathering in explosive breeding events in woodland pools still partially covered with ice, a timing advantage directly linked to their freeze tolerance. This capability has allowed wood frogs to extend their range far beyond what would otherwise be possible, becoming the only frog species in vast regions of Alaska and northern Canada where no other amphibians can survive the extreme winter conditions. The cellular mechanisms underlying freeze tolerance extend far beyond the simple production of glucose. When freezing begins, wood frogs activate specific genes that produce specialized proteins with two critical functions. Antifreeze proteins help manage ice crystal formation, while ice-nucleating proteins actually promote controlled ice formation in safe locations. This controlled ice nucleation prevents the damaging spread of ice crystals that would otherwise rupture cell membranes. These frogs also undergo extensive preparation of their cell membranes before freezing. The composition of membrane lipids changes significantly to maintain fluidity at low temperatures, similar to how fish in Arctic waters modify their cell membranes. Membrane proteins are modified to continue functioning in high glucose environments that would disrupt normal protein activity. Even the mitochondria, the cellular powerhouses, undergo structural changes that enable them to rapidly resume energy production upon thawing. Another crucial adaptation involves managing oxidative stress. When oxygen flow resumes after thawing, it creates potentially damaging free radicals in tissues, a phenomenon called reperfusion injury that's a major concern in human medicine. Freeze-tolerant frogs produce elevated levels of antioxidant enzymes that neutralize these harmful molecules, preventing the damage that often occurs in oxygen-deprived tissues during recovery. This aspect of freeze tolerance has particular relevance to human medicine, where reperfusion injury remains a significant challenge in treating stroke, heart attack, and trauma. Perhaps the most remarkable aspect of freeze tolerance involves the brain. Neural tissue is typically extremely susceptible to both oxygen deprivation and physical damage from ice crystals. The human brain begins sustaining damage after just minutes without oxygen, yet freeze-tolerant frogs protect their brains during freezing periods that can last months. These frogs have evolved specialized neuron membranes with unique compositions that resist cold damage. They also dramatically increase production of protective proteins that prevent programmed cell death under stress conditions. When most animals experience severe oxygen deprivation, neurons trigger apoptosis, essentially cellular suicide, but freeze-tolerant frogs suppress these pathways. Their brain cells also undergo structural reorganization before freezing, with changes to synaptic connections and ion channels that allow them to survive in a dormant state and rapidly reestablish neural function upon thawing. Even more impressively, these frogs retain their memories and learned behaviors after thawing. Studies have shown that wood frogs can remember spatial layouts and learned responses after being frozen solid, demonstrating that their neural protection mechanisms preserve not just the physical structure of the brain but its functional information storage as well. This comprehensive neural protection represents one of the most sophisticated aspects of freeze tolerance and one with significant implications for understanding and potentially treating human brain injuries. The exceptional cryoprotective abilities of freeze-tolerant frogs have attracted significant attention from medical researchers worldwide. Understanding how these amphibians protect their cells and tissues during freezing could revolutionize organ preservation for transplantation. Currently, human organs remain viable for transplantation for only hours outside the body, severely limiting the geographic range for organ sharing and creating critical time pressures. Researchers at the University of Minnesota and other institutions are studying wood frog cryoprotectants to develop better preservation solutions for human tissues. The mechanisms that protect frog hearts during freezing may be particularly valuable, as heart tissue is especially susceptible to oxygen deprivation damage. Beyond organ preservation, these adaptations could inform cryonic preservation technologies or treatments for conditions involving ischemia, restricted blood flow that damages tissues. Some scientists speculate that understanding how frogs prevent cell death during freezing might eventually contribute to suspended animation techniques for long-duration space travel or medical emergencies requiring metabolic slowdown. The pharmaceutical industry has also taken keen interest in these frogs' natural compounds. Several research teams are investigating whether synthetic versions of the frog's cryoprotectant molecules could be developed as therapeutic agents for preventing tissue damage during cardiac surgery, stroke treatment, or organ transplantation. These applications remain largely in research phases, but the potential medical benefits of understanding freeze tolerance could be transformative for human medicine. As climate change alters winter conditions across North America, freeze-tolerant frogs face new challenges that test the limits of their remarkable adaptation. Increasingly variable freeze-thaw cycles and unpredictable winter temperatures may push these amphibians beyond their physiological capacities. Each freeze-thaw cycle requires significant energy expenditure as frogs produce glucose and repair any accumulated damage. More frequent cycling could deplete energy reserves before winter's end. Research is showing that southern populations with less robust freeze tolerance may be particularly vulnerable to these changing conditions. Scientists are actively monitoring how these species respond to altered winter patterns. Some populations show signs of adjusting their cryoprotective mechanisms to accommodate new conditions, while others appear more vulnerable. The very adaptation that allowed these frogs to survive extreme cold may now determine whether they can persist through rapidly changing climate conditions. The geographic variation in freeze tolerance may prove crucial for species survival, as genes from northern populations could potentially spread southward, introducing enhanced freeze tolerance to populations facing new winter challenges. This ongoing natural experiment demonstrates how evolutionary adaptations developed over millennia now face unprecedented rates of environmental change, testing the limits of biological resilience and adaptation. The freeze tolerance of North American frogs represents one of nature's most extraordinary adaptations, a testament to the remarkable plasticity of life in the face of extreme environmental challenges. These seemingly humble amphibians have essentially solved problems that continue to baffle human medical science: how to preserve complex tissues during freezing, how to prevent damage during oxygen deprivation, and how to successfully revive an organism after extended metabolic arrest. What makes this adaptation even more remarkable is that it evolved not in a specialized, rare species, but in common woodland frogs found across the continent. The wood frog demonstrates that sometimes the most revolutionary biological innovations aren't found in exotic tropical species but in common creatures that have evolved extraordinary solutions to survive in extreme environments right beneath our feet. Their ability to temporarily suspend the basic requirements of life, heartbeat, breathing, brain activity, while maintaining the potential to fully restore these functions challenges our fundamental understanding of the boundaries between life and death. By studying these remarkable amphibians, we gain not only scientific knowledge with practical applications but also a deeper appreciation for the ingenious adaptations that enable life to persist in seemingly impossible conditions. A powerful reminder of nature's boundless capacity for innovation in the face of extreme challenges. The remarkable freeze tolerance of wood frogs, spring peepers, and gray tree frogs represents one of nature's most profound biological achievements: the ability to temporarily suspend life itself and later resume it without harm. Through complex biochemical adaptations refined over evolutionary time, these amphibians transform their bodies into living cryopreservation systems, protecting their cells, organs, and even memories through months of being frozen solid. Their sophisticated strategies for managing glucose production, controlling ice formation, protecting cell membranes, and preventing oxidative damage offer revolutionary insights for human medicine, particularly in organ preservation, stroke treatment, and trauma care. As climate change presents new challenges to these freeze-tolerant species, Their response may demonstrate the limits and possibilities of biological adaptation in real time. These common woodland frogs, through their extraordinary survival mechanism, remind us that the boundary between life and death can be more fluid than we once believed, and that solutions to some of medicine's most challenging problems may be hopping through the forests of North America, carrying within them secrets perfected through millions of years of evolution. Thanks for watching. Please like, subscribe, and hit the bell icon for updates. Your feedback is appreciated. We hope to see you again.